Go ahead.
Thank you. Thank you very much, Ola, for this kind introduction. And I'm very happy to give a presentation here today for all of you. So I have a lot of slides on really basic interferon work, and I hope I can also show you some of the work I'm doing here in the lab. And we already had a presentation in December from Professor Dittmar, where he was already showing some of my data, but we will talk a little bit more in general about interference. So I will first share my screen. Let's see. No, that's the one. Okay, can you see my screen right now?
Yes, we can.
Okay.
Nice.
Let's take laser point and delay. Okay, so as Ursula already said, I'm interested in interference and viral infections, and I will start with an experiment which was done in the 1950s by these two guys here. So they did a very nice and clear experiment. So what they did, they took chicken embryo fibroblast and they took a virus. In this case here, it was influenza virus, and they heat inactivated the virus. So it means the virus cannot replicate anymore. And they gave this heat inactivated virus to the chicken cells. And then they took the supernatants of the cells, and this is just marked here, and put this to other chicken cells, and came in with a virus, again, an influenza virus, which was not heat inactivated. So this means this virus can still replicate in the cells. And what they saw is a complete protection of the cells. So they called this phenomenon viral interference, and therefore the name of the group of interference came from. So just that the virus here itself, or the heat inactivated virus, produced any soluble molecule which could be transferred to new cells and transferred this protection. And nowadays we know that there are 3 different types of interference, and depending on the receptor they are using, they are either type 1, 2, or 3 interference. So we have numerous type 1 interference. So there's alpha and beta, epsilon, kappa, and omega. And I heard you're all medicine students, so then you're maybe familiar a little bit with interferon alpha, because that's also used in the clinics. There's the group of type 2 interference, where there's only interferon gamma. And there's a third group, which is quite new, let's say, and this is a group of type 3 interference, interferon lambda. And there we also know lambda 1, 2, and 3. So I'm only interested in type 1, also a little bit in type 3, but I will completely focus today on type 1 interference. And there's also a talk by Mirko Trilling, I don't know if this week or next week, and he's more interested in type 2 interference. So I think you will also get to know some basics on type 2 interference within the next days. So what are these type 1 interference? So we say they are immediate host response to viral infection. So when you have a viral infection, within minutes or let's say within hours, type 1 interference are produced. And how are they produced? So the viruses, but also other pathogens, they are recognized by so-called pattern recognition receptors. They can be toll-like receptors, but also RIG-I-like helicases or other sensors within the cell or on the surface on the cell. And whenever they recognize motifs of viruses or bacteria, so these are conserved motifs, then they activate a downstream signaling cascade, and at the end there's the production of type 1 interference. And these type 1 interference, they can then act on the same cells, so autocrine, or they can act on neighboring cells, so this is then called paracrine, via binding to their receptor. And the receptor is called interferon alpha beta receptor, and it consists of 2 subunits, 1 and 2. And all these type 1 interference, they have various biological activities. So the main function is the antiviral activity, but also in tumor therapy they are used. So they are also anti-proliferative, but they can also influence the immune response or the effector response of immune cells. So, and this is how the signaling looks like. So we have here again these three different types of interferon, type 1, type 3, and type 2. And you see that the type 2 signaling pathway is completely distinct from the others, whereas type 1 and type 3 interferon pathway, we have the same downstream signaling pathway, but we have different receptors on the surface. And one big difference between these both is the expression of the receptors. So the receptors for type 1 interferons are expressed on all cells within the body, whereas type 3 interferons are only expressed on endosomal cells, so on distinct cell subsets. So this is not expressed on all cells in the human body. So what we then see is, so here in gray, the gray dot here is the interferon. It binds first to FNR2, and then the other subunit, FNR1, is translocated here, and they form a so-called trimer. And therefore, we have then the activation of the downstream signaling pathway, which is called the JAK-STAT pathway due to the kinases here, JAK, and then we have the STAT molecules. They form then heterodimers, STAT1 and STAT2, and together with this factor here, the interferon regular factor 9, they enter the nucleus of the cell, and they bind to a specific motif in the DNA, and this is called the interferon stimulatory response element, short ISRE, and it will be also important at the end of my talk. And this induces then numerous so-called interferon stimulated genes. So 100 of different genes which are stimulated when this pathway here is activated. So what does it mean? We have these hundreds of interferon stimulated genes. And the first thing is what they do is they induce a so-called antiviral state in the cell. This can either be a cell which is already infected by a virus. So there are a lot of different genes transcribed which have a direct antiviral activity. So some are shown here. They can directly inhibit viral translation or viral transcription or replication. So I will not go here into details, just that you see there are so many different genes or proteins which can directly interfere with viral replication. But the good thing is also the cells around the infected cells, they can also see the interferon and they start to upregulate all these proteins, meaning that then they are somehow in an alarm state for incoming virus. So they are, let's say, better occupied when a virus comes in and can directly inhibit viral replication. So this is called the antiviral state. The other thing what interferons can do is they can modulate immune cell function. And I think Gennady Zelinsky already told you yesterday a lot about CD8 T cells, maybe also CD4 T cells, regulatory T cells, which are also shown here. And interferons, as I said, the receptor is on every cell. So they can, they also have an influence on the effector function of almost all cells. So when we look here for CD8 T cells, which are also important for me, they can increase their effector function and they can inhibit their proliferation. They also have an effect on their survival and they increase their cytotoxicity, what maybe also Gennady yesterday told you, granzyme production, perforin production, and also for CD4 T helper cells, type 1 interferons can further induce in production of interferon gamma, which has also a direct antiviral activity, and especially for dendritic cells, which are important to really initiate an effective adaptive immune response, so to initiate CD8 T cell responses or CD4 T cell responses. The interferons can increase the cross-presentation, they leads to maturation of the dendritic cells. Then they also upregulate co-stimulatory molecules like CD80 or CD40, which are important for activation of the T cells. They induce the production of IL-2. And they also have effects on macrophages or also on NK cells. So also on NK cells, the production of interferon gamma is increased by interference, but also the cytotoxic molecules like granzymes and perforins are increased after interferon treatment. However, they can also have kind of negative or regulatory effects. And two factors which are known as PD1 and PDL1, which are upregulated by interferon alpha. And I think maybe Gennady also talked about this yesterday, about PD1, PDL1, these exhaustion markers, and also IL-10. This cytokine is also known to be upregulated or by interferon alpha, and IL-10 is also known to more dampen immune responses. It's a more inhibitory cytokine. So with this, you would say, wow, interferons are so good. So they are strongly antiviral. They can improve the immune cell function. So they improve the immune response against the virus. So that should be very good. And then you have an incoming virus, and after a few days, it should be eliminated. But this is not the case. I mean, viruses have evolved many different strategies to really evade this interferon immune response. And what they can do is they can, on the one hand, inhibit the interferon production. And this is from an older paper. It's already 15 years old, but it's still, this is still true what's written here. So they can either inhibit the receptors which sense the incoming virus that the interferon is not produced, or they have an effect on the downstream signaling cascades here of this toll-like receptor signalings. Or another thing is that they can also inhibit the signaling of the interferon. So maybe there is interferon induced during the infection, but the downstream signaling is inactivated, what we see here from a lot of different viruses. And I mean, especially from SARS coronavirus 2, we also know right now that the induction, but also the signaling of the interferons is strongly inhibited by SARS coronavirus infection. So, and what you can also see that for some viruses, you see here, for an example, herpes simplex virus, you see that it inhibits the interferon signaling at different stages. So it also inhibits here, but it's also inhibiting there, so that at the end, you really have an efficient downregulation of the interferon response. So when we think let's say this is very basic, a very basic cartoon, let's say, of a time course of a viral infection. So what comes first? What comes then? So first, very, very early, we have the interferon response. So this is the innate response, which is then followed by antibodies, IgM. Then we have, after a few days, the cytotoxic CD8 T cells, which are coming then. And then later on, we have this very specific antibodies, IgG. So especially the CD8 T cells and the IgG antibodies, these are more belonging to the adaptive immune response. So the question is, what happens when we take away the interferon response? So if the interferon is not efficiently induced, or this downstream signaling is completely abolished, so what happens? And for this, there's a mouse available. This is called the IFNA knockout mouse model, where this mouse do not express the IFNA1, so one part of the receptor on the surface. And so what means is that then the complete downstream signaling is inhibited. So you do see an induction of interferon in these mice, but these mice, they cannot respond to the produced interferon. So due to the knockout of the receptor, so there's no signaling cascade here, and therefore not the induction of any interferon stimulated genes. And there's a nice review from 2016 where they showed what does it mean when the receptor is missing in different viral infections. And they looked here for influenza virus. At that time was SARS-CoV-1 infection, West Nile virus, the LCMV, this lymphochoriomeningitis virus. You will also hear about this this week or next week. The vesicular stomatitis virus or Vaccinia virus. And what you see that in all of these different cases, the viral replication is increased when the mice do not have the receptor on the surface. So indicating that, yes, there is interferon produced, but the interferon itself cannot do anything on the virus if the receptor is missing. So you need the receptor for the activation of the downstream signaling cascade, and so the transcription of all these interferon stimulated genes, which then either directly inhibit the virus replication or transcription or translation, or they improve the host immune response that T cells and K cells, macrophages, can efficiently fight against the virus. So, and then you also see, like here in West Nile virus, you see that also the mortality and the morbidity is increased when this receptor is missing. Also the same for vesicular stomatitis virus or Vaccinia virus, indicating the importance of the type 1 interferon or cytokine family for the protection against different viruses. So that's really important. So what some of you might know is that interferon is also used as an antiviral drug in the clinics. And there human interferon alpha 2 is used, mainly together with another antiviral component like ribavirin to treat chronic viral infections. Nowadays only hepatitis B virus infection, but in the past also hepatitis C virus infection was treated with interferon. And there are also some cancer entities where interferons are still used, like polycythemia vera or renal cancer, also AIDS-associated Kaposi sarcoma. In melanoma, it's also used and is an adjuvant therapy. And this is the structure here of interferon alpha, and you can see it has 5 helices. Yes, and as Ursula said, I'm biochemist, but we will not go too deep into the structure. But I only want to show you is that nowadays in the clinic, you're not using the natural cytokine as it's shown here. You're using a pegylated form. This means that this is first expressed. Sorry? This is any question? No. Okay. This is first expressed in bacteria and E. coli, and then later on they translationally modify the protein with this molecule. This strongly increases the size of the interferons, and this then finally leads to a longer half-life time. So, and this is shown here. So when you take interferon, which is not pegylated, you have a half-life time of 6 hours. So it means with the unpegylated form, the patients, you have to inject it into patients every other day. And when you have this new interferons, which are pegylated, then here the half-life time is increased up to 40 or 80 hours. And then you have to give this to patients only once a week. And with the new form, this Besremi, which is really, really new, you only have to give it every 2 weeks to patients. So this is really good. So what happened with interferon when you give it as an antiviral drug? And this is very important. And this is here from interferon therapy in hepatitis C virus patients. And what you see is when you start with the treatment here, you see a first phase where we have a very strong decline in viral loads. This is shown here, HCV viral load, and this is very strong. And this is due to this antiviral efficacy of the interferons, what I just explained to you. So we have all these different interferon stimulated genes which are expressed, which directly inhibit viral replication, the translation, the formation of new virions, and so on. And then you have a second phase in the interferon therapy, which is much slower. And here, And this is due to the immunomodulatory effect of the interferons. So the interferons improve the effector functions of mainly CD8 positive T cells, so the cytotoxic T cells. And in the case of hepatitis C virus infection, they have to, at the end, clear or eliminate every infected hepatocyte. And this takes a lot of time, but therefore here the slope is much slower. But this is also working. And this is really important when you're thinking about an interferon therapy. It's not only antiviral, you always have these different phases. And you see, especially, I mean, it's only a cartoon, but here you see that with this direct antiviral effect, you only increase the virus by factor 10. So this is just here 1 log less, whereas down here, the response of the immune system is at the end, it takes much longer, but it's much more effective. So you have here more than 2 logs, so factor 100 decrease in the viral loads. And this is also something what we see in anti-tumor treatment. You have a very strong anti-proliferative effect of the interferons, meaning that they inhibit the proliferation of the tumor cells. But they also improve the effector functions of NK cells and CD4 T cells that they go to the tumor environment and start to clear the tumor cells. So we can maybe do a pause here if there are any questions. Otherwise, I will move on.
No?
No? Okay.
I think there was one question right behind you. The lady right behind you.
We have one.
Your consideration using interferons to prevent and treatment COVID-19?
In COVID-19, so in COVID-19, there were different studies, and there they used especially the interferon as in a nebulized form. So they use it as a spray, because it has to go to the lung. It's a good question which concentration they used there. I mean, I know what we use in the cell culture. I know what people use for this Pegasus or for PEG intron, but which concentration they use in the spray in SARS-CoV-2, I don't know. I'm sorry.
Okay, thank you.
Okay, so then I will go on. So I already told you a little bit that type 1 interferons, they have a lot of good characteristics, but a few years ago, there were 2 studies showing that interferons can also be quite bad. And these studies are in persistent LCMV infection. So this is a virus which is only infecting mice. But this is more a model for chronic viral infection. So I think some years ago, it was a model for HIV infection and hepatitis C virus infection, but let's say it's a model for chronic viral infections. So, and both of the studies, what they did is they infected mice with this virus. And here you see they waited 10 days, 20 days, and 30 days. And so these are all normal mice. And what they did is in the red bars here, they injected an antibody which blocks interferon. So this is an anti-IFNA1 antibody, meaning that the interferon which is produced cannot bind to a receptor, now downstream signaling. So what they see in the beginning of the infection, so day 10, here you have a higher viral replication when the type 1 interferon is blocked. So they said, okay, yeah, during acute infection, it's really important to control the virus. However, what they saw at later time, but especially if you look here at 30 days, so we are 4 weeks post-infection, that those mice which do not get this antibody, so where the interferon downstream signaling pathway is still working, they had much higher virus compared to the other ones. So it seems here that if you block interferon in a later time point, that's more an advantage for the host. And if you have the interference here in this chronic infection, it's more a disadvantage. That was really surprising, because in most of the cases, the dogma was, okay, if you take out the interference, then it's really bad for the host. And here they saw that it really depends on the timing of the infection. If you look early, there, the interferon response is really crucial. And if you take it out there, then the virus replicates much more here. However, in the later phase, it seems that if you take out the interference, it's much better for the host here. And they saw this in very different tissues here: spleen, liver, lung, and serum. That was really interesting at that time, and this is now 10 years ago. And then 2 years later, there was the same group did a new paper, and what they investigated the same. So they did the same experiments, but now they used the same antibody as before, where they blocked the whole receptor. But then they took 2 other groups of mice, where one group only got an antibody against interferon beta, and the other one against interferon alpha. And I told you in the beginning that to the type 1 interferon is a big family. You have alpha, you have beta, omega, epsilon, and so on. But also beta is a very crucial cytokine of this family. And what they saw, and this is shown here. So when we only look at the late phase, again, we have the ones with the control antibody. When you block the IFNA, as the same on the slide before, you have a strong decrease in viral loads, indicating that it's really a disadvantage for the host here to have interferon around. And what they saw then is when they block only alpha, interferon alpha, this has no effect, as it's shown here. However, when they block interferon beta, then they can nicely see that this is the effect which they also saw with the receptor. And the same was true in all the different tissues. Why do I tell you this? Because this nicely shows you how different these interferons are working. So especially alpha and beta are completely different in their biological activity. And maybe you know, for multiple sclerosis patients, there you're giving those patients interferon beta. And why is it like this? What they saw when they blocked interferon beta here, that for the early viral infection, you really need interferon alpha. This is important to control the early viral replication, but it has no effect on this long-term viral effect. However, when you block interferon beta, what they saw is that you improve the T cell response. So they had here much more antiviral CD4 and CD8 T cells, and this leads then that leads to a stronger clearance of the persistent virus. And also, what they could also show is when they block interferon beta, that they had less dendritic cells which are infected compared to when interferon beta is around. And this was then finally the effect, that when you block beta, you have a much lower viral load compared to the infection alone. That was really interesting at that time, because everyone thought alpha and beta are doing the complete same thing. And this was one of the first studies really showing that, yes, they are binding to the same receptor, and yes, they are activating the same signaling pathway, but they are really doing different things. That was really interesting at that time. And I mean, now it becomes more and more complicated, because we don't have not only interferon alpha and beta and kappa and epsilon and so on, we have a lot of interferon alpha subtypes, and this is something what we have in every vertebrate. So if you look in humans, in mice, in dogs, in cows, you find always these different interferon alpha subtypes, which are highly homolog. So they share sequence homology from between 75 to 99%. And these are here the human ones. So there we have 13 genes. So there's one gene for alpha 1 and alpha 13, but there are only 12 proteins. So the genes are different, but at the end, the final product is the same for 1 and 13. So we have 12 different interferons. And if you go in mice, we have 14 different interferons. And if you go in pigs or bats, the numbers are quite different. And this is also shown here. If you look here, like for pigs, that's 18, cows have 13, horses have only 6, and bats are 3. So that's really, really different. You don't find them here in frogs or zebrafish, so it's really in vertebrates. There you can find all these different type 1 interferons. So what do we know about all these different interferon alpha subtypes? So, I mean, this is similar to the slide I showed you before. So we have the interferons here that bind to their receptor. We have the activation of this downstream signaling, and then we have the induction of the interferon stimulated genes. So what we know so far is they all have different affinities to their receptor. So indicating that especially interferon alpha 1 is a very bad bind or weak binder. It's really bad in binding to receptor subunit 2. These ones here are much better, and these ones here are the best. So alpha 10, 17, 6, 14, and 7, and so on. So these are really, really tight binder. What is also an interesting or important fact is how is the receptor expressed on the surface and how is this regulated. So there are some studies from hepatitis C virus infection, but also from some autoimmune diseases, where they see that this one subunit, if not two, has also two shorter forms, and one of these shorter forms is a soluble form. So meaning that maybe here the expression might be influenced by any inflammation or virus. So we have looked in HIV infection. There is no effect on the receptor expression, but also the receptor is differently expressed if you look in a B cell or an NK cell or a T cell. So especially on B cells, there's really a lot of the receptor on the surface, whereas on T cells, it's much lower. Then what is also quite interesting is this is not the only pathway which is activated. There are also a lot of other signaling pathways which are activated by interferons. And then we have really a wide pattern of different interferon stimulated genes, and these then orchestrate the biological activities. So is it more antiviral? Is it more changing the immune response? Is it more antiproliferative? And so on. So what we have done in the past, and these were really, really basic experiments in the beginning, we were interested in the role of interference in friend virus infection. I think you already heard about this virus yesterday. Yes? Yeah. So I don't have to say so much about it. But what is important for you to know is that friend virus is very, very bad in inducing type 1 interference. So it also has an inhibitory factor which really downregulates the interferon response, that at the end, if you look during the infection, you can never measure any interference in these mice. So when you take the plasma of the mice and do an ELISA, you cannot really measure an induction of type 1 interference during friend virus infection. What you can see is you have like a slight increase when you look in the spleen cells. You see, yeah, there are some interferon stimulated genes upregulated during infection, but it's not a really strong interferon response. So what we did, And the first studies here is we started with an interferon treatment with these different subtypes. And here we use the mouse subtypes. And this is my focus in the lab. We are producing them in the lab, and then we are testing them in a lot of different models. So we started here already one day prior to infection, but it's also working when we start later. So we start with the treatment with the interference, and then we infect those mice with the friend virus, and we do this treatment for, I'm sorry for the kinetic here. So there's something wrong with the numbers here, but we did that for 10 days, and then we sacrificed the mice, and we were interested what is the effect of the different interference. Because I mean, this was already done when I came to the lab in 2007, as Ursula said. And also at that time point, everyone thought, yes, of course, we have a lot of different interference, but they're all doing the same. That was the dogma at that time point. So what we did here, we did 2 different studies. In the first one, we tested 4 different interference, and then we tested 3 more, and they are shown here, where we tested interferon alpha 2, 5, and 11. This is not the human alpha 2, which is used in the clinic, so it's all mouse, and you cannot compare mouse with humans. They are completely different. But you see that interferon alpha 11 could significantly reduce the viral loads here in the spleen, whereas 2 and 5 didn't work. So we also then, we were always asked, then just give the mice 10 times more, 20 times more of the interferon, and then it should work. And we tested that, but it didn't work. It had no effect. When we injected 20 times more interferon alpha 2 or interferon alpha 5, these mice still could not control friend virus infection. What was quite interesting that this effect was really long lasting, because I mean, especially from HIV infection, when you treat patients and you stop treatment, then you directly see viral rebound. So the virus is coming back, and for us that was really good. So we did this treatment in the acute phase of the infection, and still 5 weeks later we could see a significant difference in the viral loads. Yeah, so that was really, really surprising for us. And then, of course, I told you the immune responses are very important, and interference can modulate immune responses. So therefore, we had a closer look on the T cells and NK cells. We did not see any effect on T cells. That was really surprising for us. So then we started to work on NK cells. So these are these natural killer cells which belong to the innate immune system. And what we saw is that they are strongly activated. So this is CD69. It's an activation marker for NK cells. And you see that friend virus only moderately activates the NK cells compared here to the uninfected mice. But this was strongly enhanced when we treated the mice with interferon alpha 11, but not with 2 or 5. And when we then depleted these NK cells during the treatment, so this is shown here. So again, we have the friend virus infection. We treat them with alpha 11. You see this very strong induction in viral loads. When you only inhibit NK cells during friend virus infection, there is no significant decrease. So in the normal infection without interference, they are not having such an important role, especially during 10 days post-infection. But what happened when we deplete, so we take out the NK cells from mice which got the treatment here, and there you see that the strong antiviral effect is partially gone. So you see there is still a reduction compared to the untreated mice, but not as strong as with the mice which do harbor NK cells. And this means, again, like I showed you before, for hepatitis C virus infection, you have these 2 different effects of the interference. You have a direct antiviral effect, which is still working here. I mean, the NK cells are gone, but these mice can still express these interferon-stimulated genes, so they can directly inhibit friend virus replication. But the second part, this efficient immune response, this is missing here. And this effect is gone here. So it's really like half of the response, so 50% is really missing when we take out the NK cells. We then also analyzed them in more detail, and we could see that especially here granzymes, so these cytotoxic molecules were strongly enhanced or strongly expressed here on NK cells, and also trail is another cytotoxic ligand on the surface of NK cells, which binds to a so-called death receptor on target cells. This is also strongly upregulated here compared to the friend virus only mice. And this is also shown here for all mice we tested. So that was really, really interesting for us. So this was the acute infection. We had this strong effect of interferon alpha 11, whereas 2 and 5 had no effect. And that was also the case that people were asking, yeah, this is really strange. Interferons are doing all the same. Also alpha 2 and alpha 5, they should also activate NK cells, but this was not the case here. We were then interested in the acute, in the chronic infection, and there we started to treat the mice. And here we wanted to compare interferon alpha and interferon beta. I showed you before that interferon beta can also have a more negative role on the immune response. So therefore we tested both, and we used here alpha 11 as one interferon alpha, and of course beta. We first tested them in vitro, so we infected just cells with friend virus, and those were working, here alpha 11 and beta. And then we tested them in the acute infection, so we treated mice and then looked after 10 days, so similar to the results before. And also here, both treatments, alpha 11 and beta, significantly reduced viral loads. So in the acute infection, both could really suppress viral replication. We could not really see interferon alpha in the serum of the mice, so that was not surprising. But then we went in the chronic infection. So we infected again these mice and let them sit for 6 weeks, around 6 weeks. And then we treated them again 5 times with alpha 11 and interferon beta. And that was again very interesting, because similar to the results I showed you in LCMV, interferon beta itself had no effect on the viral loads, whereas treatment with alpha 11 significantly reduced the friend virus infection. We could also show that we do see a lot of interferon stimulated genes which are apopulated after the treatment with alpha 11, but this was not the case for interferon beta. And we also saw here that during chronic infection, interferon alpha 11 has a very strong effect on the cytotoxicity of CD8 T cells. And this is also very interesting. This is also very interesting because you see that in the acute infection, this cytokine is mainly modulating NK cell responses, whereas in the chronic infection, it's mainly modulating the T cell responses. That was really interesting. If you are maybe more interested in tumor response, then especially anti-proliferative properties of interferons are really, really important. And what we did to analyze those effects is we performed an assay where we had dendritic cells from a mouse. We put an antigen on it, and then we cultured them together with the CD8 positive T cells, which recognize the specific antigen. And then we did the different interferons there, and then we were interested in what's going on with the proliferation of the T cells and the effector functions. And we labeled those cells with some dye. We used here violet tracer, as it's called, so they are purple than the cells, let's say. And whenever they proliferate, so they divide, they give half of the dye to their daughter cells, and when they proliferate, they again give half of the dye to their daughter cells. And so when you look in the via flow cytometry, you see, and this is like how it's looking when cells are not proliferating. You have one one peak here, which is high in this dye. So the cells have not proliferated. However, when the cells have proliferated strongly, you see these different peaks, which are shown here. And the lower here, the concentration of the dye, means the higher is the proliferation rate. So, and this is what we see here. So when we look in those cells which do not see interference, you see that they strongly proliferate. So we have here this, here's the correlate, here's the everything right from this stretch here did not proliferate, and all these cells here, they proliferated very strongly. And this is how it looks like. And you see that for some interference, like here for alpha 9, for alpha 6, for alpha 4, the proliferation was strongly inhibited. And this is also seen here. So we see that those cells which do not see interference, they strongly proliferate. However, when you add different interference like alpha 2, 1, and all the others, you do see an effect on the proliferation, but it differs very strongly depending on the interferon we use. So especially alpha 4, 6, 9, those were really, really strongly inhibiting the proliferation of these cells. And apart from their direct effect of the proliferation, we were also interested in their effector functions. So do the interferon treatment or the stimulation with the interferons also changes their cytotoxic capacity or cytokine expression and so on. And to see already for granzyme, they all express high. So here, high percentages of cells are already positive for granzyme. However, if you look inside the cell, you really see that the interference itself strongly increase the expression of granzyme B, which was much, much lower compared to those cells which haven't seen interference. This means that, yeah, the stimulation, this co-culture, which is also shown here, induces granzyme B expression in almost all cells. However, those cells which have seen interference, they start to produce more granzyme. So you don't have more cells which are positive or which are expressing granzyme, but within the cells, you have higher concentrations of granzymes. And what you can also then see is that especially those interference which strongly inhibit the proliferation, like 4, 6, and 9, they can also strongly kill target cells. So this is an assay where you look where you check the the cytotoxic potential of these CD8 T cells. So we just take these cells and let them kill some target cells. And there you see that the untreated cells only killed like 20% of the target cells, whereas those cells which were treated with interference, they killed up to 60 to 80% of the target cells. So the especially here, this increase in granzyme B expression strongly improves their killing capacity. This was mouse stuff, and but I'm also interested in human infections, and this is maybe also more familiar for you. And one model we are working on is HIV infection. And what we did some time ago is we got some blood from healthy individuals. We isolated the white blood cells, so the PBMCs. We put them here in cell culture. We infected them with HIV and treated them with human interference. And then we waited some days and looked for viral replication. And this is how it looks like. So when we had here the untreated cells, we normalized this to 100% infection. And then we had a look at the different interference. So this is interferon alpha 2, which is also used in the clinics. You see 50% reduction in infection. However, there are other interference like alpha 6, alpha 14, 17, 21, which are much, much more potent in inhibiting HIV infection. And also in the past, there were a lot of different clinical studies where interferon alpha-2 was also used in HIV patients. There were really a lot of studies, and in almost all studies, there was only very, very moderate effect on the outcome of the interferon therapy, but in most of the cases, there was almost no effect of the interferon alpha-2 therapy. And this might explain that interferon alpha-2 maybe just not working against HIV, whereas other cytokines like 14, 6, 17 can really efficiently block interferon alpha HIV infection. You may ask right now, why are people using interferon alpha-2 in the clinics? This was just the first interferon which was recombinantly produced. So that one was the first which people produced in bacteria, and then all the science started with interferon alpha-2, and at that time point they didn't even know that there are so many interferons. They knew some but not so many. So that started in the early 90s, and that's the reason why only alpha-2 is up to now used for clinical treatment. And also all the patents are all on alpha-2, and all the studies all with alpha-2. So these are data in vitro, and if we are working with HIV, you cannot really use good mouse models because you cannot infect mice with HIV. I mean, of course, you can work with chimpanzees or rhesus macaques or anything, but this is also for us in Germany quite difficult, and we don't have a monkey facility here. So therefore, we worked with so-called humanized mice. So these are mice where the complete immune system is changed by human cells. This was done. So the people, we did that in collaboration with people from the US, because this is also not allowed in Europe. So they used mice which do not express which have knockouts in very, very efficient or important genes. So RAG is important for T and B cells, for the T cell and B cell receptor expression. Gamma C is important for all these cytokines, IL-2, IL-12, IL-6 for their production, and CD47 is a very important, very famous macrophage marker. So they use these mice where all these markers are missing. So these mice do not really have a good adaptive and innate immune system. And then these mice were lethally irradiated, and then they got a little bit of fetal liver, fetal thymus, and bone marrow cells. So hematopoietic stem cells. Therefore, these mice are called B, L, and T mice. So you let them sit for 12 weeks after the transplantation, and then these mice have a complete human immune system. So if you take out some blood and you measure them, all immune cells are human. And then you can start to infect these mice with HIV or HBV or every human virus you want to use. So what we did then, we infected these mice with HIV because now they have the human CD4 on the surface, the T cells, and also the co-receptors, so CCR4, CXCR5. CXCR4 and CCR5, sorry. So these mice were infected with HIV, and then we started the treatment. And here we used the two interferons, which were the one which were the most effective one, alpha-14, and then we took one which is used in the clinic, so alpha-2 as a comparison. And what we saw is when we treated those mice, here HIV positive mice with interferon alpha-14, all mice were below detection limit when we looked for viral antigen. Here, this is capsid antigen of the virus P24. You can just measure this in the plasma. And when we treat the mice with interferon alpha-2, yeah, there are some mice where it was reduced, but it was not significant. So in most of the mice, we had comparable viral loads as in the untreated mice. And the same was true so this is a viral protein. The same was true if you look for viral nucleic acids. There you also see for our viral RNA here, HIV RNA for interferon alpha 14, that was all below the detection limit, except these two here. But again, for alpha 2, this was not working at all. I deleted one scale here. So we did then the same experiment again. This is missing here. I don't know why. And we just stopped the treatment after 10 days, and then we let them sit for another 10 days. And we killed the mice at day 21 post-infection, just to see if this treatment interruption has any effect on the viral loads, as I showed you in friend virus infection. And yeah, unfortunately, and this is what you can see here, at 11 days post-infection, this is this time point, we see this very low viral loads in these alpha 14 treated mice. However, when we just let the mice sit for 10 days, do nothing, there is a complete viral rebound. So virus again starts to replicate, and after 10 days, it looks the same compared to untreated mice or compared to alpha 2 treated mice. So that was not so nice. But then again, we were interested in the immune responses. And if you think about HIV infection or also hepatitis B virus infection or hepatitis C virus infection, many people are talking about chronic hyperimmune activation. So it's controversially discussed that there are interferon expressed during chronic infection, which induce a chronic hyperinflammation. So what we did is we looked for the activation of CD4 and CD8 T cells. This is one marker in humans, HLRDR. And you see there was no effect on the activation of the T cells. So no hyperimmune activation. That was really good. And then we had a closer look on the CD8 T cells, on NK cells. And what was quite interesting is that for interferon alpha 2, and this is shown here for alpha 2, we saw that it specifically modulates CD8 T cell response. So you see here more granzyme and also more CD107A. So this is the marker which is upregulated when granzyme is leaving the cells. But alpha 14 mainly targets NK cells. So there again, this ligand here, which targets the death receptor, was upregulated. That was really surprising that here in one mouse, in the mouse, you really see one interferon is mainly going to T cells, the other one is mainly going to NK cells. That was really astonishing. That was in acute infection, and then we said, okay, let's go in a chronic infection, because I mean, if you go in a clinic, then patients are usually coming with a chronic HIV infection, and you do not really see the patients with an acute HIV infection, so a few days after getting infected. So we used the same mouse model, infected those mice. Oh, I'm sorry, this is Germany. Yeah, these weeks, and we let them sit for 6 weeks, and then we start the treatment. So we gave them some mouse chow where was an antiretroviral therapy inside, which is mentioned here. And then we also had one group which received an interferon treatment, interferon alpha 14. So the idea was that if we combine this antiretroviral treatment with interferon, where we can also nicely activate the NK cell responses, we can mainly also reduce the viral reservoir during HIV infection. And this is what you see here. So yes, we do decrease viral loads when we treat the mice only with antiretroviral treatment. However, the combination further reduced this effect. And this was really, really nice. So also the therapy in a chronic infection and HIV was nicely working. I think now let's see. So this was HIV. Let's go to HBV.
Maybe some questions, Katrin?
Yeah, sure. If there are some questions, sure, we can also stop. I mean, if not, then we will just move on, and I can answer the questions. There is one. Okay, perfect.
We have one question.
I have a question. May you deeply explain why interferon do not completely inhibit tumor cells, or we can possibly eliminate tumor cells?
It's a very good question. I mean, one thing what you also see in patients is that the interferons also have very, very severe side effects. The problem is that the receptor is expressed on every cell. So if you give interferon to patients, every cell can respond to interferon. And this means that not only a T cell can be activated, every cell in the body can be activated by interferons. And then, of course, also tumor cells, similar to viruses, they have also adapted different strategies to evade this immune response. So, and there are also different tumor proteins can downregulate the interferon system. So also tumors have maybe lower expression of the receptor on the surface, or the downstream signaling cascade is downregulated, then that the interferon cannot really work so efficiently. So that are the reasons why at the end the interferons are not completely eliminating the tumors. But I think also one problem is that only interferon alpha-2 is used in the clinics. And we know here from many clinicians here that especially in hemato-oncologic cancers or diseases, interferons work really, really good. They cannot really explain why it's working so good, but there in many, many patients, they see really good results when they give interference to the patients. But at the end, it's not working in all of the patients. That might also depend on the sex of the patients, the age, if they're smokers, non-smokers, if they're taking any drugs or alcohol abuse, and so on. So that's really something at the moment what we are not, what is not completely understood, but this is something we are working on. But this is a really, really good and really important question. Thanks.
Thank you, Franz.
Okay, let's go to HPV infection. And there also.
Sorry, I think there's one more question.
Oh, okay, sorry.
Different interferons are responsible for the inhibition of different parts of the viral DNA in the process of replication. So, are there some mechanisms that are controlling amount of output of different types of interferons? Can the organism understand which interferon is better to produce against the virus present in the cell, or maybe this is genetically controlled process?
Really good question. So, I mean, this is at the end what I'm doing every day. I want to understand why these interferons are so different, and at the end to figure out which interferon is the best against which virus or against which tumor and so on. And this is something what we have to study right now. So what we do is that if you, and I have some data at the end, I'm not sure if we will manage this due to time. When we look in SARS-CoV-2 infection and we treat cells with different interferons, you see complete different gene expression pattern. And this is at the end really important, and that depends on the virus, what you have, but also on the cell type where you're looking. But finally, we have to somehow understand how these interferons are working and why are they differentially working on a T cell compared to an NK cell, compared to a liver cell that's completely different, or compared to a lung cell. And if we do understand, then we can maybe manipulate the interferons, maybe create new interferon molecules. That's also something we're doing in the lab, which with stronger antiviral activity or stronger immunomodulatory activity, more targeting T cells or NK cells, or we can maybe combine different interferons for a therapy. Yeah, this is a really, really a good point. But yeah, at the moment, this is something I cannot completely answer you this question. So we definitely have to work more on that. But this is the main focus of my work here in the lab.
Okay, thank you.
You're welcome. Okay, then going back to HPV. Perfect. Well, thanks, Ursula. So, and especially in HPV, I mean, you should know that, I mean, of course, it's a virus, but it's also an oncogenic virus. So it can induce a tumor, hepatocellular carcinoma at the end in the liver. And therefore, it's really important for us to work with interference in HPV, because there you really have the effects on the immune system, on the virus, but also on the tumor development. And this is still the only viral infection where interferons are used in the clinics. And there we know that around 30% of the patients nicely respond to interference. That's what the literature said. But there also we know that depending on age, sex, and so on, and also the type of HPV, the serotype, the outcome of the therapy strongly differs. So we first started with the mouse model, and this is a model where you inject a plasmid encoding for HPV into the mouse. Here it's the same. You cannot just infect mice with HPV because mice do not express a receptor for the virus. So we use the model where you inject very high volume into the tail vein of the mice in a very short period of time, so a few seconds. And then you have kind of a liver disruption, and this results in a transfection of the liver with the plasmid. And this plasmid encodes hepatitis B virus, and so then it somehow mimics an infection. You can nicely use this model for really looking into immune responses against this virus. So what we did is, so we used this hydrodynamic injection with the HPV plasmid, treated the mice. So now we have the mouse interference, and looked at different time points for delivery for HPV markers, serological markers. So we looked for HBs antigen, HPV DNA, and also core antigen here. And what we can see here is that, especially, and this is shown here in blue and in green, especially interferon alpha 4 and 5, they strongly inhibit HPV replication in the mouse. And this is interesting because before I showed you data in the friend virus infection mice, and there interferon alpha 5 did completely nothing. And there you can nicely see now we are in the liver. Before, friend virus was mainly spleen and the blood system, and now we are in the liver. And you have complete different outcome of the interference. So from the first data, we'd say take alpha 11, it's always the best. Now look here, alpha 11 is not working at all. It's here similar to the mice which do not get any interferon. So it really depends on the virus and also the tissue which is infected. So this is also really important. And then, of course, from the mouse system, we went in the in vivo situation. So for this, we isolated hepatocytes from somehow healthy. So we call them healthy, but I mean, they have any kinds of tumors in the liver, but they are not HPV infected. So we isolated the hepatocytes, and then you can nicely infect them with hepatitis B, and we treat them with interference. And I just also did that today here in the lab. I got hepatocytes from Spain because it's very difficult to get those cells, and we got them today from Spain, and I just infected them today. And then tomorrow I will start with the treatment with the interference. So then we keep that for 9 days, and then we analyzed it. And this is what you see again. Alpha 2 is shown here, is the one which is clinically used, and similar to HIV, you see 50% reduction. But again, you see that others like 6 or 14, also 17, are much stronger. And this is shown here for one serological marker, HBe antigen, but the same is true here for HPV DNA, and also for the circular closed, covalently closed circular DNA here, cccDNA, which resides in infected cells. And there we also saw that alpha 14 could downregulate the copies per cell. And there was something which is really interesting, and I don't want to go so much into detail, but I told you that interference can also activate a lot of different downstream signaling events. And I showed you that the main pathway, and I can just show it here again, is here STAT1, STAT2. This leads then here to binding here to this ISRE, and then we have the transcription of the interference stimulated genes. However, what you can nicely see here is the treatment with alpha 14 and not with alpha 2 strongly activated STAT3 pathway and also STAT6 pathway, indicating that in contrast to interferon alpha-2, alpha-14 can also activate other pathways. So this is really important. And then what we could also show is that especially interferon alpha-14 not only uses this pathway, it also uses this pathway. So and this leads then here, this is here the ISRE element, and here this means it binds just to another part of the promoter regions, so to other regions in the DNA, and this leads to the transcription of other genes. And this is something what we can see with interferon alpha-14 here in purple. So you see that this pathway here is strongly upregulated, what you can see here, but it can also activate this pathway, which is normally only activated by interferon gamma. This was really surprising, because this was not the case for alpha-2. If you look here for alpha-2, it activates this pathway here, but it's not activating the second pathway here. So here we have really an individual activity of interferon alpha-14 in hepatocytes. We are right now also looking in other cells if it can also activate this pathway also in other cells, but this is ongoing work. I don't have the data yet, but this is really interesting. And this was, and then we did the same experiments, and we combined interferon gamma, which is, when I just go back, which is then activating this pathway, with alpha-2, which is activating this pathway. And if we combine these alpha-2 and gamma, we have the same effect like when we only use 14. So indicating that here really alpha-14 have a really, really strong effect by activating these two different pathways. And this is also seen, I know it's a busy slide, but this is also seen when you look for RNA. These are transcriptomic data, so they look for RNAs which are expressed, and you see that there's one gene here, GBP5, and this is a gene which is, if I go back, mainly expressed via this pathway. And this is only expressed here, strongly expressed by 14, but not by alpha-2. The same here. So this was really, really important that we see here for the first time that interferon alpha-14 uses two different signaling pathways which are not used by alpha-2 alone. Now we go in, but maybe I will skip that a little bit, and maybe very short, nor only in SARS-CoV-2, because this is maybe also important for you. So what we know about SARS-CoV-2, and I will just shorten that a little bit, is that the virus itself is also a poor inducer of type 1 interferon. We know, like right now with Omicron, we do see a little bit more interferon, but with the original strains and the other variants of concern, there's only a low induction of type 1 interferon, and especially because the virus expresses a lot of proteins which directly evade these pathways. There's like ORF6, but there are also others. But in experiments in vitro have shown that the virus is very sensitive to type 1 interferon. And I will, and maybe what you have already heard is that doing life-threatening COVID-19, so especially patients with a very severe disease, up to 30% of those patients have autoantibodies against type 1 interferons. And this is something what we can also see in patients, in hepatitis B virus patients, so that these patients develop antibodies against type 1 interferons. And what means this? That when they have these antibodies here, they cannot really, so the interferon which is produced is then blocked by these antibodies, and they don't have an efficient expression of the interferon stimulant genes. This is really important. And there is a hypothesis that we have, especially in severe COVID patients, we have either patients which have inborn errors of type 1 interferon, which is just shown here. So they have like a morphism in some genes which are important for interference, like IR7, TLR3, or the receptor IFNA1, or you have these patients which express the autoantibodies. So this is really a huge part of these really life-threatening diseases. And it means that in those patients, especially in the acute infection, you do not really see a good type 1 interferon response because it's either not induced or you have these autoantibodies which are induced in these patients. And so this, they cannot really, the interferons which are produced only at a low level, but they cannot really work because they are neutralized by these autoantibodies. And then with this very insufficient type 1 interferon response in the beginning, then the virus is growing and replicating and so on. And this results at the end that you have a really very strong antiviral infection. You have damage of the immune system and a really systemic inflammation. And this is what we see in COVID-19. So let's go a little bit further. So what we have done, we used a cell model. These are kidney cells from African green monkeys. This is a nice model for SARS-CoV-2, and we infected them with different interferons here. And again, we saw some were good, some were not good. And here we saw that especially alpha-4 and alpha-5 were really good in inhibiting SARS-CoV-2 infection. And also we had lambda here, lambda-3 as a control because that was already published. This was really good. And you also see it here. So we did immunofluorescence and we stained the infected cells in green. And you see here when we treated the cells with alpha-5, they're almost only 2 cells are still there. Whereas when we treat them with another one like alpha-16, it's the picture is really greenish, so there are a lot of infected cells. And so we separated them into different groups of high antiviral, but then we have low to moderate antiviral and almost no antiviral activity. So this is a cell line, so this is not the best cell culture model, so therefore we use really human cells from the blood. And this is what you can see here. Because the problem with these cells here, they cannot really produce their own interference. So they lack the interferon system. And therefore we used this human airway epithelial cells. So we got that from lung transplant patients. And then you take the cells in cell culture. You can keep them for a few weeks. They differentiate and they really look like the cells later on in the lung. And so this is how they look like. And this is called an air-liquid interface because when they're fully differentiated, you keep, let's say, the bottom is in media where you have the liquid, but the top here is in the air. So that's really kind of like in the lung. And we used the cells and infected them with SARS-CoV-2 and did the same experiments. And there you see the same. So you see that interferon alpha-4 and 5 strongly inhibiting SARS-CoV-2, whereas others here, like 1, 6, 16, did not really had a response on that. We then looked for their gene transcription, and this was just a question we got before. We really wanted to figure out what is the difference there. Why do we see differences? Why is the one interferon working, the other one not? And these are here just genes, and we did some analysis where they just tell you these genes belong to antiviral immune response, these belong to translation, and so on. And what you see is that especially those genes here which are high antiviral, you see there are a lot of dots here, indicating that there are a lot of genes are expressed, and you also find genes which are not expressed like in the other groups here. Like here for alpha-5, we see genes which are important for signaling pathways or lymphocyte activation. And when we have a closer look at these antiviral immune response, because this is the one, I mean, we were just in cell culture without any immune cells, so we were looking for the antiviral immune response. There you can nicely see. So these are standard interferon stimulated genes, which are shown here. And there you see that, especially here for some of them, and this is quite interesting, you see that those interferons which are really high antiviral, they strongly induce those, whereas the others, you see the color is more in blue. also downregulate these. This was really, really important. And just this is my final slide. And then again, we go in a mouse model, in a humanized mouse model, but these mice only have humanized human fetal lung transplants, which were here. So they were transplanted, and then after 12 weeks, you can infect this human lung transplant, and this is shown here with SARS-CoV-2, and we treated those mice with the interferons to really see if the effects which we saw in vitro, would they also work in vivo. And again, we used alpha-5, which has the strongest effect in vitro, and alpha-2, which is the clinically approved one. And so these are only a few mice, but that was the first experiments with these so-called lung-only mice, where only the lung is human. You see that we do see significant downregulation here. These are 3 logs, so factor 1000 reduced when we treat the mouse with alpha-5, which was not the case when we treated mice with alpha-2. So coming to an end, so I hope I could convince you that so we have a lot of different interferon alphas, alpha subtypes, but there's also type 1 interference. It's a very heterogeneous group. We have, and all these different interferon alpha subtypes, they have, of course, similar functions or overlapping functions, but they also have unique properties. And I showed you this slide, and they can induce a new unique pattern of ISGs. So that's something what we especially saw in SARS-CoV-2 infection, what I showed you here a little bit, but we have also seen that in HPV infection, like in GBP5, which was only expressed by alpha-14, but not by alpha-2. They have very distinct antiviral activities. I mean, if you remember the mouse data for HPV, especially 4 and 5 were really good, whereas in French virus, so retrovirus infection in the mice, 5 wasn't working at all. So that's quite interesting. And they also differ in their immunomodulatory potency. So if you remember the data from HIV, so there we saw that alpha-2 is mainly activating the CD8 T cells, the cytotoxicity of them, whereas CD14 was mainly activating NK cells. And they also vary in their anti-proliferative activity. With this, I would like to end. And I just want to announce that we have a new workshop here, which will be held here in April, and I'm organizing it with 2 of my colleagues here. And I mean, if you're happy if you're interested in HIV, then it would be also nice if you want to join us for our workshop. If you have any questions concerning the workshop, you can always write me an email. And ask me, and I can also send you the flyer if you want. And with that, I'm at the end, and I thank you. And I'm open for some more questions.
Thank you so much, Katrin.
Yeah, thank you very much for the lecture. Can we ask one question, please?
Sure. You can also ask more. I have time.
Thank you very much. What you can tell about traditional treatment with interferon using treatment of influenza? What you can tell about it?
Oh, with influenza.
Is your opinion about it, please?
So I know from mouse studies that interference in influenza are very, very different. So we know from mouse studies that really depending on how much virus there is there inside, at which timing it is, the interference can be either beneficial or also really bad to the host. So I think in most of the cases, I think that for really an acute infection, which are very, very short, where the virus replicates only for a few days, the interferons are maybe not the best choice as a clinical treatment. It's more like in HPV, HCV infection, you need time. I mean, especially in hepatitis B virus infection and then also in hepatitis C virus infection, these treatments last for almost a year. So patients were given interferons for 48 weeks because you really need this very strong immunomodulatory effect, and this takes a lot of time till this is induced, especially in humans. So I would say that for an acute influenza infection, it might not be an therapy option. But maybe for other chronic diseases, that especially also for maybe chronic respiratory diseases, that might be really an option to treat those patients. Yeah. But there also we need to figure out what's the best subtype for the treatment and so on. And also we have to reduce the side effects. This is really important.
Thank you very much.
Erika.
So please use the opportunity. Even if you have some difficulties with English, no problem. Just try to raise the questions.
I mean, otherwise, whenever you're interested in interference and lab work, you can also contact me for an internship or anything where we are open for students. So, yeah, just contact me.
So I think you could get a really good insight about what is possible in science and what kind of opportunities there are here in the lab of Katrin and her colleagues for doing some research. We are just at the start of our Essen and Poltava cooperation, and of course we are also trying to get some funding for joint research. So this is what we are just starting, and well, I hope that there will be lots of opportunities. And also, Katrin just mentioned the HIV workshop in April here in Essen. We have just handed in an application to the German Development Agency. And this is also included in this application, that we try to get some funding for 2 or 3 people who are really interested in coming over and taking part in this workshop. So this is still pending, but we are trying to get this funding. So yeah, I hope for a lot of more opportunities in the future.
Yeah, thank you very much for your assistance and support and very interesting lectures. I think it's a very good beginning for our future cooperation and our joint research. Thank you very much.